mitochondria are very ancient organisms that are in part devoted to energy production as ATP. And a particularity of these mitochondria is that they have their own genome, the so-called mitochondrial DNA. And this genome is carrying 37 genes, of which 13 are coding for enzymatic subunits of the respiratory chain. And this mitochondrial DNA maintenance is under the control of the nuclear genome, and over 1,000 genes are known to be mandatory for mitochondria. And more recently, as Andrew mentioned, there were some evidence that maybe mitochondrial signals modulate the nuclear genome expression, even if these signals are poorly known. So a particularity of mtDNA is its presence as several copies in a mitochondria. And for instance, in sperm, you have a dozen of mtDNA copies, and in our site up to 1 million copies. And this explains the maternal inheritance of mitochondrial DNA and of mtDNA mutation. For instance, when a de novo mutation occurs in a somatic cell, due to the random distribution of mitochondria during mitosis, you can obtain various proportions of cells that could have either one only population of mtDNA, the so-called homoplasmy, and also a majority of cells that are heteroplasmic. And over threshold of mitochondrial DNA mutation, that depends both on the nature of the mutation and on the tissues. So the tissue is damaged, and then clinical symptom occurs. During oogenesis, Andrew mentioned, the unique features of mitochondrial DNA segregation also may result in various proportion of mature oocytes, either wild-type homoplasmic or mutant homoplasmic or heteroplasmic. And it's very difficult to predict the different proportions of these oocytes in mitochondrial DNA mutation carrier. And so this is why the genetic counseling of mtDNA disorders is so difficult. So briefly, some examples of the most frequent mitochondrial DNA disorders with a very difficult acronym as MILAS, as Leber hereditary optic neuropathy, NARP, and so on. And most of these disorders are really serious and without any effective treatment, so that a couple at risk of transmitting such disorders are very interested in reproductive options. So, as mentioned, Andy, nuclear genome transfer procedure aimed to achieve wild-type homoplasmic embryos in females carrying mtDNA mutation, and the method, as described by Andy, is to transfer nuclear genetic material from oocytes or pre-implantation embryos retrieved in a carrier to enucleated oocytes or pre-implantation embryos donated by a mitochondrial DNA mutation-free individual. And the purpose is to get embryos, healthy embryos, that are both devoid of mtDNA mutation and having the nuclear gene pool of their parents. So these procedures as emerge as an option, as reproductive options, due to some drawbacks, real or supposed, of the PND, prenatal diagnosis, and PGD, pre-implantation genetic diagnosis procedure. And the first of them was that assessment of mutant load in one blastomere or in CVS or amniocytes would not be an accurately reflect of the overall mutant loads in child at birth. We showed that the mutant load assessed from one human blastomere accurately reflects the mutant load of the whole embryo, and furthermore remains stable throughout the embryo fetal development. We showed that the mutant load assessed in a given fetal tissue accurately reflects the mutant load in all fetal tissues, and we also showed that the mutant load assessed in cord blood cells at birth reflects the mutant load measured previously in other fetal tissues, and irrespective of the nature of the mutation. So, as previously mentioned, it's difficult to predict for mtDNA carrier the proportion of mutant-free oocytes or oocytes with a low level of heteroplasmy. Very few scientific data exist. And I briefly present the data of three labs, the lab of Becler, Necker, and the data from David Thorburn's lab in Melbourne and Patrick Chinnery's lab in Newcastle for the two most common mutations. So for the first one, NARP, you see that for a carrier, the probability to have wild-type homoplasmic embryo is around 30%, but it's very few number of embryo or site. While for Miller's mutation, the probability of having wild-type homoplasmic fetus is around 6%. But when you take into account the probability to have a very low heteroplasmic fetus, below 30%, that is possibly predictive of a good postnatal outcome, you know that there is still very different between the two mutations, with nil probability in this mutation and around 70% probability in Miller's mutation. So, another objection is that the predictive value of the embryo-fetal mutant load for the postnatal outcome would be poor. So our attitude relied on availability of postnatal data that reported mutant load threshold for disease expression. And so, for the most common mutation, this threshold is around 60%. When you have a mutation rate that is below 60%, you're usually healthy. And only when the mutation excesses 60%, you are ill. So, with our colleague of Antoine Becler, we decided to offer to couples some option of PND or PGD based on the mutant load determined in either blastomeres or CVS or amniocytes. And below 30%, we say it's okay. Over 60%, we say it's poor prognosis. And between these two values, we discuss with the couple. So we have carried out around 53% PND, PGD, only 10 PGD. And we tried to follow up the children born after a PND or a PGD procedure. On the left panel, that is Mila's children, and on the right panel, NARP and MERF and ND subunit of complex 1 children. And in the x-axis is the level of mutant load, and the y-axis the age, and each bar depicts a child. So for this moment, from 53%, 53 acts of PND-PGD, we got 26 living alive children, and all are, for this moment, healthy. So the success rate is around 50%. So there are nevertheless real drawbacks for this procedure. The most frequent being the frequency of high levels of embryo-fetal heteroplasmy that usually result in termination of pregnancy or the impossibility of embryo reimplantation. There is also a residual risk of developing a disease in children with a low level of heteroplasmy, and especially in mutant homoplasmic women, the probability to have a healthy child is very low. So other approaches recently emerge. Some attempts of cytosolic transfer from an embryo to another one, to my knowledge, didn't give any good result for mtDNA disorders. And so the nuclear genome transfer procedures have been developed, and Andy described, reported this procedure that include either pronuclei transfer between zygotes carried out in mouse, in human, or MST between oocyte, either in monkey or human, and more recently, polar body transfer in oocyte. So, and he has mentioned the main issues. I remind you some of them. So, how to select the recipient, oocyte, or embryos? Should we select oocyte embryos from relatives, or alternatively not from relatives? What is the rate of success of such procedures? It's not known. For its moment, it doesn't seem to be high, but I imagine that it's going to improve. So, Andy mentioned also the impact on development. I mentioned the disruption of physical links between nucleus and cytosol that are poorly known, impairment of the nucleus mitochondria crosstalk, and also tolerance of the contaminant heteroplasmy, since when you are picking up a nucleus, you are exposed to artifactual transfer of mutant mitochondria DNA that are then located in the recipient donor. And it seems that the contaminant heteroplasmy might be rather low. So I don't come back to the ethics and psychological problems that are real, and no more on the legal problems and particularly prohibition of genome modification impacting the Johnson line in human. And so, I have finished. Thank you.